All right. So the first topic at hand is linkage, and the second topic that will naturally flow from it is linkage mapping. Linkage mapping is going to test your attention. Linkage is easy. Taken together, I treat it as one continuum of the topic. And in this one continuum of the topic, these are the questions that you will eventually need to answer. These are subparts, the facets of the chapter. And up until now, in the last 30 years, civil service and IFS combined, these have been the facets which have been touched. Some of the facets have not even been touched as yet. But you can be reasonably sure that sooner or later the examiner is going to touch these facets also in the question paper. Remember, the PYQs are very, very important, but they account for only 50% of the questions that you are going to see. So there is no substitute to break the syllabus into relevant question-worthy facets and then align the entire coverage of the chapter so that we at least understand those facets and we at least gather the important facts and information on those facets. And of course, there will be other connecting concepts that we will be learning, but perhaps those connecting concepts will have no significance in terms of examination question. But to make sense of everything that you are learning, you will be needing those connecting concepts also. But here there are 10 or 11 facets. First and foremost, what is linkage? You will be surprised, whole lot of students are unable to clearly define what is linkage. So what is linkage and what is the correct explanation of linkage? Second, how was linkage discovered? And in this, you are going to talk about the Betson and Punnett experiment on sweet pea, that is Lathyrus odoratus. They were basically carrying out experiment on a dihybrid cross. Flower ka color dekh rahe the, they were looking at the color of the flower and they were looking at the shape of the pollen. And when they basically checked both these things, they realized that they are not getting Mendelian results. That is 9331, they were not getting. And the result that they were getting, they were very, very hard to, they found these results very hard to explain. We will learn what made these results very hard to explain and what was the pattern here. So discovery, 10 points. So in bracket, what I have mentioned is not the marks. By now, you already know it. I am basically saying that these many points should be there with you. So what is linkage and correct explanation? 10 points. Discovery of linkage, 10 points, including the experiment. So basically 4, 5, 6 points of description and there will be one long diagram. Then types of linkage, 10 points are very easy. Then characteristic features of linkage type of inheritance, 8 points will be good enough because it is never going to be asked alone. It is going to be like, what is linkage? Enumerate its characteristic features and mention how it can be detected. So what is linkage is one part, characteristic features is another part, detection of linkage is going to be another part. And combining each one of these, you can easily generate a 20-point answer, and that is what maximum UPSC can expect from you. So the fifth facet is detection of linkage. The sixth is what is the significance of linkage, or for that matter, how does linkage influence the inheritance? Seventh part is going to be relation with crossing over. What is the relation between linkage and crossing over? So that is the seventh part. And then the remaining parts are about linkage mapping. First and foremost, what is linkage mapping and what is the underlying principle behind in linkage mapping? Next one is going to be the two types of linkage mapping. One is called the 2-point test cross, basically dihybrid test cross ke through what kind of linkage mapping we can do and what can we do with 2-point test cross is basically about two characters. So yes, dihybrid test cross. And then 3-point test cross means trihybrid test cross. Means we are considering 3 genes at once and then we are trying to map those 3 genes onto the chromosome. If you do not know yet what is chromosome mapping and all that, we are going to learn that also, don't worry. And finally, what are the benefits, what are the limitations of linkage mapping, what do we gain out of linkage mapping in the era of molecular mapping, and then we are going to see what are the limitations because of which molecular mapping is going to be the thing. Okay, so these are the things. Those who have joined late, good evening once again. Like Nikhil has sent a message, login में problem हो रही थी नहीं, लेकिन निखिल वो एक बार Zoom में free account create कर लो, and that account you can use every like time. So बस, और वो free account ही रहेगा, you don't need to pay Zoom anything. We will pay Zoom for holding these meetings longer than 45 minutes and all. You don't have to do anything. Just एक बार create कर लेना. And you have not missed, no, not Nikki. So you have not missed, Nikki, anything big. अभी हम लोग बस facets को discuss कर रहे थे, तो first 5-7 minutes आप दोबारा play करके देख लीजिएगा, ठीक है ना? Now, let us start with what is linkage and what is the correct explanation for linkage. And for this, mentally you will have to go back to the days when genetics was a science in its nascent phases. Okay, so you need to go back to that phase when genetics was a new thing and nobody knew how many genes are there. Nobody knew, for that matter, how many chromosomes are there in the human nucleus. So we didn't have much information at that point of time. हम तब की बात कर रहे हैं. So in the beginning, people believed that every 2 genes that you study will be assorting independently. Means the segregation of, let's say, gene A has nothing to do with segregation of gene B. Simple as this. So when there is a dihybrid F1, which is heterozygous for both the genes A and B, so when it comes to produce the gametes, it will produce all the 4 types of gametes. In other words, all the 4 combinations of A and B will be seen. You know that in gamete there can be only one set of chromosome. Therefore, in the gamete there can be only one allele, right? So, one type of gamete will be this, where both A and B are present in the dominant. Then another where A is dominant but B is recessive. Another is where A is recessive and B is dominant. And another one where they both are present in recessive. And when these 4 set of gametes, all right, when these 4 set of gametes meet another such 4 gametes, that is from the female parent, then any combination is possible. What do I mean? This AB can fuse with this one, it can fuse. अगर इसके साथ नहीं फ्यूज करेगा तो इसके साथ फ्यूज करेगा, करेक्ट? अगर इसके साथ नहीं करेगा तो इसके साथ करेगा, इसके साथ नहीं करेगा तो इसके साथ करेगा. And the same applies to this one also, this one also, and this one also. And when this happens, what do we see? We see 9, 3, 3, 1. That is the classic Mendelian dihybrid ratio. But when the scientists started to see a lot of other dihybrid crosses, they soon realized that all of them are not following this pattern. All of them are not following this pattern, and in some of the cases, we see the parental combination being repeated in F2 a lot. What do we mean here? Let us go back to Mendel's Pisum plant only. And for a moment imagine please, that 2 traits which Mendel studied, they were not independently assorting. Just imagine, Although Mendel studied independently assorting traits, but for a moment imagine that Mendel, the traits that he studied, were not independently assorting and they were linked. And अगर ऐसा होता तो क्या देखते? Let us say that the plant is tall and it is producing round seed, and it is mated with plant which is dwarf and that is producing wrinkled seed. So we all know that in F1 we will find heterozygous plants, but because heterozygotes are also going to show the trait of only the dominant parent, so well in F1 generation you see what, all tall, all producing round seeds. But imagine that they are linked genes. Then what will happen? That you will find tall round, you will find dwarf wrinkled in great numbers. There will be some recombinants, agreed. There will be some recombinants, means tall and wrinkled will be there and dwarf and round will be there. But in Mendelian independent assortment, let's say that tall round, dwarf wrinkled, wait a moment, tall round, ठीक है, tall and wrinkled and dwarf and round, अगर ये Mendelian होता तो the ratio had been 9:1:3:3 because these two are recombinants, so 9:3:3. This is homozygous recessive, so 9:3:3:1. But if, let us for a moment imagine that they are linked genes, what you are going to see is that tall round and dwarf wrinkled will together account for almost what? Maybe 80% of the offsprings. Okay. And this will be kind of 20% or sometimes even less. Right. So what do you see here? You see that parental type are much more abundant compared to the recombinant type. If you see, and we are going slowly around that, if you see in independent assortment, 9 is dominant parent type, 3 is recombinant, another 3 is recombinant 2, and 1 is what? 1 is parental type 2. This is how it looks like. Now if you see, so parent 1 and parent 2, they account for 10 out of 16 offsprings. Parent 1 and parent 2 account for 10 out of 16 offsprings, right? So if you now look at it, so this is slightly above. If you now look at it, it is slightly above 50%. But that is pretty much all. 10 out of 16 is slightly above 50%. But if you see 70 to 80% parental type and 20 to 30% recombinant type, then there is great likelihood that what you are seeing is an example of linkage. So now how do we define linkage? Let's try to define linkage. Linkage is basically when alleles of different genes do not assort independently because they are located on the same chromosome. Let's say that there is a gene P, there is a gene Q, recessive, and most of the gametes will be having this combination of P and Q. Most of the gametes. Why do they have this combination of gametes? Because they are located on the same chromosome. Had P been on a different chromosome and had Q been on a different chromosome, and had they been segregating, you would notice that P is making this combination also. P is making this combination also. P is making this combination also, and this combination is also possible. But if they happen to be located on the same chromosome, the greatest likelihood is that they will inherit in this combination only, and the other one will be inheriting in this combination only on the homolog. Are we getting the point? So we define linkage as a phenomenon in which the inheritance of two genes does not show independent assortment in a Mendelian way. What is Mendelian way? That is full independent assortment, which will eventually lead to 9:3:3:1 ratio. But if two genes are located on the same chromosome, they are not going to display that. Simple as this. Because during meiosis, one chromosome will inherit as one unit. And if one chromosome inherits as one unit, so a gamete, if it receives this chromosome, so it will have both P and Q in the same manner as it was with the parent. And thus, in the offsprings, you are going to see that parental type are very much dominating. So this phenomenon is called genetic linkage. Let's see the definition statement. Genes linked close together on the same chromosome are called linked genes. They belong to something known as linkage group, and linked genes travel together in the meiosis. Why? Because they are located on the same chromosome, and then they arrive in the same gamete. And since they arrive in the same gamete, so what happens that in the offsprings also, the parental combination will be predominant. And they do not assort independently in a Mendelian fashion. Even if crossing over sometimes breaks the linked genes, yet the Mendelian ratio is never touched. Mendelian ratio is possible only when the two genes that you are studying are located on separate chromosomes. This is linkage. Now, before we move any further, and मैं थोड़ी हिस्ट्री बता देता हूँ. One of the first cases was reported in sweet peas, right? Give me a moment. In sweet peas, that is Lathyrus odoratus, by William Bedson, Saunders, and R.C. Punnett. The year was 1905, very shortly after the rediscovery of Mendel's work. And what they did, that they went for crossing purple flowers and long pollen grains with red flowers and round pollen grains. And the results deviated significantly from 9:3:3:1 ratio. You would have gotten 9:3:3:1 only if the genes would have been located on different chromosomes. But since they were not located on different chromosomes, so they tended to inherit in parental combination. Here and there, yes, parental combination was broken. Broken by what? Crossing over. But it was not enough times broken that it will give you result like Mendelian independent assortment. So that is linkage. Unfortunately, Bedson and Punnett were unable to explain what is the reason behind linkage. They thought that during the meiosis, during the gamete formation, the chromosomes break. And as the chromosomes break, their segments tend to combine together in a manner it was present in the parent. So they tried to explain that chromosomes will fragment, and then fragments of the chromosome will combine and reassemble the chromosome in more or less similar configuration, similar manner as it was present in the parent. But of course, when scientists studied meiosis, during which the gametes are produced, no such breaking of chromosome was even observed. So obviously that was out of question. Another thing that they said, that some of the dominant genes have affinity for dominant allele of other gene. They call it coupling type of linkage. And then they said some of the recessive alleles have affinity for other genes recessive alleles, so they will inherit together. That was also coupling type of linkage. And then they said some of the dominant will attract recessive allele of some other gene, so that will be called repulsion. But neither coupling and repulsion hypothesis was correct, nor the chromosomal breakage and fusion hypothesis was correct. Basically, they tried in quick succession. They gave two different models, but neither of the model was successful. What was correct explanation was simple. An organism had so many genes and very few chromosomes. Therefore, every chromosome will carry a lot of genes. And because the chromosomes do not get vertically fragmented, so whatever the genes are present in whatever allelic order, maybe A dominant, B recessive, C dominant, D recessive, E recessive, F recessive, G dominant. So in the same manner, they will tend to inherit in the gamete and then inherit in the offspring, until in some of the cases crossing over breaks this combination. But whenever crossing over breaks this combination, there will be a recombinant offspring produced. But what will be the number of recombinant? Will it be 6 out of 16? Because 9, 3, 3, 1 is 16. And recombinant 1 is 3, recombinant 2 is 3. So it is 6 out of 16. So will crossing over be taking place enough number of times that in offspring set 2, F2, it will produce 6 out of 16 recombinant? Answer is no. In F2, in case of linked genes, parental type, in most of the real world examples, they will be occupying almost 70 to 80% of offspring phenotype, and in the 20%, yes, recombinants will also be there. Now, before I move any further, I show you the experiment carried out by Williamson and Betson and Punnett, not Williamson, Betson and Punnett. Uske pehle, I want to know, do you have any question? If you have any question, now please raise hand. Don't type it. Please raise hand. It will help you articulate your question, and then I will be answering it. So it is important for this particular topic that after every regular interval, we kind of check our concept. So any question that comes to your mind, unhesitatingly raise your hand and let me know. Anything, any, kuch bhi. No question is silly. Okay, Vasvi, I'm simple hand bhi raise kar doge to chalega, but no problem, Vasvi. I will. ठीक है, Shraddha. I'm going to take your question. Keep the hand raised. That is important, so that I can take your question one after one. First, Vasvi. Go ahead, Vasvi. I have sent you an unmute.
Sir, actually the linkage concept is clear, but so the PQ example is not clear. Like
Okay.
how exactly the steps.
देखो क्या है अच्छा, मुझे श्रद्धा का भी क्वेश्चन लेने दो, देन एट वन गो आई विल आंसर बोथ ऑफ यू. Shraddha, please go ahead.
Good evening, sir.
Good evening, Shraddha.
I think the linkage definition and the concept is clear, but what is explained just before this is not clear. The coupling hypothesis, and after that, how the Mendelian genetics will change.
Okay, ठीक है, मैं no worries, no worries. Let's get this, okay? So let us understand, all of us, okay? Now you can lower your hand. So let us understand, all of us, how independent assortment takes place. So here we are. There are two chromosomes, okay? Here. there are two chromosomes, and on these two chromosomes, two different genes are present. These are two different chromosomes, okay? And there is a gene called P and there is a gene called Q, okay? And then we have to make its homolog also, because in any parent there are going to be homologous pairs, है ना? We all are diploid, right? We all are diploid. So ये तो clear है ना कि हमारे पास दो set of chromosomes होंगे. And since we have two sets of chromosomes, so we are going to have two copies of each gene. And it is very much possible that two copies are different from each other, means the person can be heterozygotic. So here it gets interesting. अच्छा, बीच बीच में मैं पूछने वाला हूं कि इज इट क्लियर और नॉट, एंड व्हाट यू आर गोइंग टू डू दैट इफ इट इज क्लियर यू आर गोइंग टू शो मी अ थम्स अप. ओके, सो देखो ये रेसेसिव अलील्स हो गए, राइट? सो इन सबको थोड़ा मैं सेंटर में मूव करता हूं, राइट? सो दिस इज वन पेरेंटल सेल, पर्टिकुलर it is a parental cell which is going through meiosis. It is going to divide. Okay. So, अच्छा एक चीज रह गया. Give me a moment. So this is one parental cell. मैं सर्कल नहीं बना रहा हूं for a reason. Now let us see कि इसमें meiosis होगी तो क्या होगा. Meiosis will separate the two homologs together, right? Away from each other. So take a look here. These two are homologous to each other. If you can see, these two are homologous to each other, and these two are homologous to each other. Correct. So what will meiosis do? Meiosis will separate the homologs. Correct. And as meiosis has separated the homologs, these cells will complete meiosis. Now, this set of gametes will be producing dominant P, dominant Q, and this will be producing recessive P, recessive Q. Correct? So on the meiosis 1 metaphase, these chromosomes were aligned face to face, and coincidentally, the dominant allele of both the genes were on one side and recessive allele were on other side. So when it was separated, it will give recessive allele gametes and this will give dominant allele gametes. But is it not possible that in some other cell, like when gametes are getting produced, so a lot of cells are going to go through meiosis. When gametes are getting produced, so a lot of cells are going to go through meiosis, not just one cell. So is it not possible that in some other cell, this is how these chromosomes have aligned on the metaphase plate? Is it not possible? Yes, no, just show a thumbs up like this. Increase the number. There are 51 people. Keep increasing. All right. ठीक है. Now lower it. It is possible. अब चल. यहां तक समझ में आ गई ना बात? चलो. अब अगर यहां गैमीट बनेगा तो क्या बनेगा? देखो, सेपरेट हो गए. ओके. अब क्या गैमीट बनी? रेसेसिव पी के साथ डोमिनेंट क्यू आ गया और डोमिनेंट पी के साथ रेसेसिव क्यू आ गया. So there is one organism. This is one organism. And when one organism is producing gametes, so there are going to be so many gamete mother cells. So from one set of gamete mother cell, you get these two types of gametes, where this was the alignment. So you get these two types of gametes. So this person made these two types of gametes. But the very same person using other gamete mother cell, like spermatogonial cells, okay, is also making this type of gamete and this type of, sorry, this type of gamete. So one person did what? Made all the four possible types of gametes. Correct? If you think it is correct, then please raise a thumb again. It looks childish, but it is important. एक ही बार में अब ये clear होने जा रहा है. And it is not childish. ठीक है, so I can understand कि most of you have understood. Okay, lower the thumbs up. Anyone who has any question, ठीक है, anyone who has any question यहाँ तक में raise hand so that I can unmute and I can hear the question and no hesitation. ठीक है. Yes, let me take the questions and then I'll move. Those who understood, please be patient. किसी दिन आपको भी कुछ नहीं समझ में आ गया तो I'll be equally patient. Okay, Vasvi and then Shiva. ठीक है, मैं अभी आ रहा हूं. Shiva Prasad, go ahead.
Hello?
Yes, Shiva.
Sir, actually, I didn't understand the last step. How did that?
देखो, Shiva, good that you asked me. In one person, there will be lot of spermatogonial cells, correct? Each cell will go through meiosis. In some of them, dominant P and dominant Q is on one side, recessive P and recessive Q is one side. So when this cell divides, so it gave rise to this and it gave rise to this. But at the same time, see, chromosome can align like this, align like this. Let me use two colors and you can understand. Is it clear? This is black. Do I have something red? Yeah. So this is red. Yeah. So take a look here. So it is possible that in one of the cell it is like this, and it is very much possible that in some other cell it is like this. And how it is physically there, it will separate in this way. And it has nothing to do with what the other set of chromosomes are doing. So all the four types of combination are possible. So in this one, let's say that recessive P and dominant Q are on one side and dominant P and recessive Q are on other side. So if there is a division, so now you will see recessive P, dominant Q, and dominant P and recessive Q. And that was one person, this one person has made all the four types of gametes. So Shiva, is it clear now? Go ahead, Shiva, and tell me, is it clear now?
Yes, sir. Yes, sir.
Is it clear? Okay. Ritika, what's the question? Ritika, you can unmute yourself and tell me. Vasvi, what's your question?
Sir, what are homologous chromosomes?
Okay. So you are clear that we have got 46 chromosomes as human beings? Are you clear, Vasvi? है ना?
Yes, sir. I'm clear.
Right. Okay. Now 23 came from mother, 23 came from father. You got it? So we have got two sets of 23 chromosomes. So these chromosomes are called homologous chromosomes. So हमारे पास हैं 23 अलग अलग टाइप के क्रोमोसोम, but every chromosome is present in two copies. So chromosomes which are present as copies are called homologous chromosomes. Are we clear? Ritika, I was trying to unmute you. Shiva, you still have any question? Else you can lower your hand. Or if you have a question, please do ask. Ritika, I'm trying to unmute you the way I unmuted other people. So now you can unmute. I'm sending you the unmute option. And if you are having difficulty, then you can simply type your question. So I'm assuming that everyone else has gotten this thing. So we can move a little further now. Give me just one moment. Let me see if there is anyone with a question. No. ठीक है, ठीक है, ठीक है, ठीक है. चलो. So this much you have understood. Now let us see कि if all the four types of gametes are getting produced, if all the four types of gametes are getting produced, just see how you get 9331 ratio. Even if it is repetition from the Mendelian genetics, but why not we be very clear about it. One set of gametes here and one set of gametes here, correct? And then first set of offsprings, second, third, and fourth. And you all know that there are all the four combinations of PQ possible, correct? So in yellow color, the gametes PQ, P recessive Q, recessive P dominant Q, both recessive. Same here, PQ, P recessive Q, recessive P dominant Q, and both recessive. And remember, if you have got one, right? So if you have got one dominant copy, that particular trait will be dominant. So in this one, please pay attention to this punnet. Ritika, I'm coming to your question in a moment's time. So please pay attention to this one. So in this one, both P and Q, okay, they both are dominant. So whatever is coming from the other parent doesn't matter because one dominant copy of P, one dominant copy of Q have become available, and as a result, other allele, it could be dominant, it could be recessive, doesn't matter. It is now going to show both P and Q in the dominant phenotype. So offsprings in pink color. So we are writing P dash Q dash, P dash Q dash, P dash Q dash. Dash means doesn't matter, it is recessive or dominant. And again P dash Q dash. Where did the dominant copy come from? From this one. And similarly, look here. So up to down also it is going to be P dash Q dash because here also a dominant copy is there. Correct? So P dash Q dash, P dash Q dash, and P dash Q dash. Then you have got this one. Here you have got a dominant P. Can you see that? So इन दोनों में dominant P ही होगा, इन सब में dominant P चलेगा, right? So dominant P. All right. And then you have got Here dominant P. So top to down also it will be dominant P and dominant P. Then here you have got dominant Q. Can you see that? So it will be like Q dash, Q dash, and Q dash. Then here you have got dominant Q. So it will be like Q dash here. It is already Q dash. What does it mean? It is homozygous dominant, and this is again Q dash. Correct. And then you have got other ones. Why not we do some counting? So those which are showing dominant of P and recessive of Q is going to be one recombinant. P dominant, Q dominant is one parent type, and P recessive or Q recessive is other parent type. But P dominant, Q recessive is recombinant. Similarly, P recessive, Q dominant is also a recombinant. So let's identify P dominant, Q recessive. Can you see that? Why not we color it with green? Okay. Then P dominant, Q recessive. We find it here. Why not we color it green? Any other P dominant, Q recessive? Here we find P dominant, Q recessive. We color it green. Are we done? Now let us find P recessive, Q dominant. P recessive, Q dominant. We will be doing it bright blue. Bright blue. P recessive, Q dominant. So bright blue. Where else we find P recessive, Q dominant? Here we find P recessive, Q dominant, bright blue. Then let us search for P recessive, Q recessive. Only one here, and we are going to select some nice and fancy color. Let's color it orange. Now take a look. What is the ratio? Now all the four phenotypes are in other color. Let's put some number. Pink one. 1, 2, 3, 4, 5, 6, 7, 8, 9. Done. Then 1, 2, 3. So 9, 3. Okay. Then we have got 1, 2, 3. So 9, 3, 3. And then we get 1. So what have you gotten? 9, 3, 3, 1. But the question is, how did you get it? That's the big question. And the answer is simple. You got it because these four types of gamete could be formed. So why did we get all these four types of gamete? You got all these four types of gametes because they were located on separate chromosome. As a result, gametes could have a line like this also, and gametes could have a line like this also. And these two could have had very, very different results. And as a result, we saw independent assortment, same individual forming all the types, same individual forming all the types of gametes, and same would apply to the female because female has also got these two genes on two different chromosomes. So female is also going to show independent assortment. And that ways you get Mendelian 9:3:1 ratio. Okay, इसको मैं ठीक कर देता हूँ. Otherwise, when I share the PDF, it will be a little confusing. Okay. So now I hope you have understood why independent assortment takes place when the genes are located on separate chromosomes. Let's imagine for two other genes, let's call them A and B. Okay. So in this case, A and B are present on the same chromosome. A, B. Let's say this is the combination. Okay. And the other homologue, I'll have to change its color, has got other alleles. So when during meiosis they separate, what will happen? They separate, and then you get this gamete and this gamete. What are your chances of getting this? Very low. What are your chances of getting this? Very low. But is it at all a chance? Answer is yes. It is still a chance, provided there is crossing over. Crossing over will mean what? So crossing over will basically, I hope you have understood this much. Let me take some question. Vidyadhar is asking, is there any relation between linkage and multi-gene families? No, Vidya. Ritika is asking, sir, does it mean that cell in one body have different orientation of chromosome? Why not? Every cell, when it goes through meiosis, means gametic cells, when they go through meiosis. So how do the chromosomes align on the spindle apparatus is entirely going to be different from cell to cell. So yes, that happens, Ritika. So I hope now you are clear. जो आपका डाउट था रितिका, है ना? चलो. So now they can still provide, they can still provide a chance to produce these gametes. But for this, crossing over has to take place. Now, what is crossing over? Here. In crossing over, what happens? Let me erase all of this. So, give me a moment. Okay, ठीक है. So in crossing over, I just wanted to enlarge it a bit. Now, in crossing over, what happens? That chromosomes will exchange. I'm right now simplifying, but chromosomes can exchange equal amount of their segments between each other. So, no, it will still not go. But let's imagine that this red chunk has gone here and this yellow chunk has come here. And in that case, you will get recessive A, recessive B together and dominant A, dominant B together. Reason being this A has gone here and this A has come here. So crossing over can still create recombinants, but not in the same number. That will compare with 6 out of 16. Is it clear now? Why do we not see that kind of ratio. Even though we find recombinants in linkage, why do we not see the same Mendelian ratio? Is it clear to everyone? Just show me a thumbs up before we move further. Nice. Okay. Please lower this. Please lower this. Please lower this. And anyone who has any question left, let me answer those questions first, and then we will quickly move to the next part. Shraddha, you seem to have a question?
So I just need to confirm that we can say that both crossing over and Mendelian genetics will give recombinants, but the percentage of recombinants which we get from crossing over is much lesser than that we get from Mendelian genetics. So is this correct?
Absolutely. इससे करेक्ट बात आज मैंने पूरे दिन में कुछ नहीं सुनी. Seriously. This is absolutely correct. You got it. ठीक है ना?
Thank you.
Anyone else with any question? Okay. Chalo. So now let's move. अब आप लोगों ने अच्छा एक बात बताओ, ऐसे पनेट स्क्वायर बनाना समझ में तो आ गया था. इस पनेट स्क्वायर में तो किसी को कोई प्रॉब्लम नहीं थी ना? जिसको प्रॉब्लम है वो बस हाथ उठा दे. ठीक है, इस तरह से पनेट स्क्वायर, क्योंकि ऐसा पनेट स्क्वायर हम आगे भी बनाएंगे. आज ऐसे लग नहीं रहा कि हम लोग लिंकेज मैपिंग कर पाएंगे, कल करेंगे, डोंट वरी. लेकिन ये पनेट स्क्वायर में तो किसी को कोई प्रॉब्लम नहीं है ना ऐसे बनाने में? अगर है तो हाथ उठा दो. कल मैं मान के चलूंगा कि सबको आता है ऐसे वाला पनेट स्क्वायर बनाना, बिकॉज इट विल बी अ बिग टाइम सेवर here and in the UPSC main examination. All right. Give me a moment now. We are now going to move to other parts. So हम लोग यहाँ पे थे. ठीक है, ठीक है, हम लोग यहाँ पे थे कि it was discovered by Betson and Punnett, and how they basically tried to interpret but could not interpret. So what was the explanation given by Betson and Punnett? Explanation given by Betson and Punnett. Let me just write it. One was called breakage and fusion hypothesis. So this hypothesis basically said that chromosomes get fragmented and later the fragments reunite according to the parental template. कि जैसा पेरेंट में था वैसे उसी को टेम्पलेट समझ के वो रीयूनाइट कर जाते हैं. बट व्हाट हैपेंड, देयर इज नो एविडेंस फॉर दिस. एंड डू यू नो दैट इन साइंस जिस चीज़ की एविडेंस नहीं होती है उसको करेक्ट नहीं माना जाता. ठीक है, सो yes, you can use, someone has asked this question, can we use this kind of Punnett square in the exam? Absolutely, this is the Punnett square जो लोग बना पाते हैं एग्जाम में और बनाना चाहिए भी. So basically this theory got discarded. Later on they attempted at coupling and repulsion hypothesis. So essentially what coupling and repulsion hypothesis said, that some gene alleles attract same type of alleles of other genes, alleles of other genes. So that was coupling, and second is some alleles attract the opposite type of alleles of other genes. So this is called repulsion. But the fact is, and listen to this thing very carefully, this is called repulsion. The fact is कि इसका भी कोई evidence नहीं है, and therefore this is also wrong. Right. And because they could not explain it correctly, so Betson and Punnett are only credited with discovering the phenomenon of linkage, but not having explained it correctly. And then came T.H. Morgan's explanation. So what did T.H. Morgan explain? Simple. There are many traits in an organism. Okay. And since there are many traits in an organism, therefore there are many genes. But there are limited number of chromosomes. And if there is limited number of chromosomes, therefore several genes per chromosome. And as a result, they will inherit mainly in parental combination. And since they inherit in parental combination, it is known as what? It is linkage. That it seems that two genes are linked with each other, therefore they will always inherit in parent type combination, and rarely crossing over may perhaps break it, but not as frequently as the independent assortment would generate the recombinants. So long story short, अगर हम chapter की beginning पे जाएं, so now what you know already is what is linkage, the correct explanation of it, discovery and the attempt by discoverers to explain how linkage takes place. So अब हम लोग जरा discovery पे जाते हैं. So this is what Betson and Punnett were studying. Okay. So this is sweet peas. That is Lathyrus odoratus. So take a note here. One set of plant had purple flowers and long pollen. This is the homozygous dominant for both the genes. And other one had red flower and round pollen, which is homozygous recessive for both of them. You carry out their crossing, and in the first generation, F1, you get all purple flowers and all long pollens, all of them. Perfect. Then you carry out selfing. And notice here, let's do some math. So you have got, give me a moment. Yeah. So you have got this as parental type, and you have got this as parental type. Correct. So 284 and 55. These two are parental type. All right. So you have basically got this number. All right. And then these are recombinant 21 and 21. So just add this number. So now what you get is this number. So out of 481, 439, right? Almost 440 are what? Almost 440 are parental type, which is almost close to 90% if you see. Very close to 90%. So 90% is parental type. Parental type will mean this one and this one. And in 10%, both the recombinants have fitted in. And now compare 6 out of 16. In Mendelian, 6 out of 16 would have been what? 6 out of 16 would have been the non-parental type. And here what you see? 10 out of 100. So this was the discovery. Needless to say, this is very, very important because if you don't know this cross, you don't need to remember this number, but ये याद रखना कि 10% recombinant हुए थे. So this is an important cross. Okay. And always remember about 10% recombinants थे यहाँ पे. Numbers आपके लिए matter नहीं करते उतना. And around 90% थे parental type. So that's what. So this is the discovery part. Now let us come to what the important types of linkage. This is easy part. This is another illustration of the same thing. Let's say if genes are completely linked means if there is no crossing over. ठीक है, देखो. So यहाँ पे what you are going to do is a test cross. And remember, if the genes are completely linked, if there is no crossing over, तब की बात है. So this is heterozygous. So normal leaves and tall plant. There are two genes, M and D. Heterozygous, okay? And this is homozygous recessive. Both the genes are recessive. Here the mottle leaf, mottle means थोड़ा सा wrinkled, और plant भी dwarf है. Let's imagine कि कोई क्रॉसिंग ओवर नहीं हो रही, तो ये तो बस यही दो गैमिट बनाएगा, ये वाली गैमिट बनाएगा और ये वाली गैमिट बनाएगा, करेक्ट? और ये बस एक ही टाइप की गैमिट बनाएगा, ये वाली. सो half of the gamete will be dominant M, dominant D, half of the gamete are going to be recessive M, recessive D, and this is going to only produce recessive M, recessive D. And then in equal ratio, half of the plant will be normal, heterozygous, and half of the plant will be dwarf and mottle leaf, homozygous recessive. So here we see कि if we carry out test cross for totally linked genes, mean genes के बीच कोई crossing over नहीं होती, so the ratio is going to be 1:1. But if it is assorting independently, the ratio will be 1:1:1:1:1. टेस्ट क्रॉस हमने मेंडेलियन जेनेटिक्स में देखा है, करेक्ट? तो अगर ये अलग अलग क्रोमोसोम पे हैं तो ऑल द 4 टाइप्स ऑफ गैमीट्स विल बी फॉर्म्ड, and here only one type of gamete will be formed, and you will get all the 4 but in equal ratio. So this is one very powerful method, very powerful method to detect linkage. So अगर कोई पूछे कि linkage को detect कैसे करते हैं, so this is detection of linkage. How? By carrying out test cross, right? So, and even if there are recombinants, they are going to be much less than equal ratio of 1 versus 1 versus 1 versus 1. there will always be predominance of parental type. So now we are done with the detection part. फीचर्स हम बाद में आते हैं, पहले हम टाइप्स पे आते हैं और फिर हम लोग फीचर्स को दोबारा देखेंगे. सो दीज आर द टाइप्स ऑफ लिंकेज. और देखो यहां पे कपलिंग और रिपल्सन टर्म वापस आ रहा है. द टर्म कपलिंग एंड रिपल्सन आर कमिंग बैक, बट नॉट इन द कॉन्टेक्स्ट ऑफ explanation, but in the context of types of linkage. Let's say, based on strength, there are two types of linkage. Complete linkage, where there is nearly no crossing over. What do we mean here? We mean that the genes that you are studying are very close to each other. Therefore, there is going to be no crossing over. Partial, there is going to be crossing over, and there is some significant distance between the genes. Therefore, the genes are essentially getting crossing over between them. Based on chromosome, sex linkage is linkage on sex chromosomes, and somatic linkage is linkage on autosomes. In the chat box, I would want someone to tell me, what do you mean by sex chromosomes? As fast as possible. Chat box में बताओ, what do you mean by sex linkage? Nana, must not X and Y. Tell me what they are. You are right now telling me in mammals, these are the two types of X chromosome. Okay. So, Shraddha is asking 6 out of 16 नहीं है. Shraddha, 6 out of 16. इन मेंडल में 9, 3, 3, 1, तो कितने ऑफ स्प्रिंग है? 16. और रिकॉम्बिनेंट कितने हैं? 6. तो मैं कह रहा था 6 आउट ऑफ 16. और बेटसन एंड पनेट में हमें क्या मिल रहा था? रफली 10%. दैट्स व्हाट आई वाज सेइंग. इज इट क्लियर? चलो. Zava is saying determining the sex of organism. Perfect. They determine the, Shraddha is saying determine the sex of the organism. Good. Which determine the sex of the organism. Good. The chromosome that determines. अच्छा, सारे लोग एक ही आंसर लिख रहे हैं. Chromosomes. Janvi, you got it wrong. Chromosomes present in gametes. No. All the chromosomes are going to be present in gametes. Abhijit is saying identifying gender. No, Abhijit, identifying sex. Sex is a biological concept. Gender is a social construct. Okay. So biological world में there is no such thing as gender. Mitali, sex chromosomes are the chromosomes that determine the maleness or femaleness. Fair enough. Sex of the organism involved in sex determination. Okay. Good. ठीक है. Most of you have got it correct. But one thing that you all miss is simple, that sex chromosomes will be carrying the master gene for sex determination, but they also carry other autosomal gene. So other autosomal genes, if they happen to be present on sex chromosome, then those genes will be called sex-linked genes. Right. So linkage on sex chromosomes, especially somatic function genes. For example, in human beings, red-green color blindness gene is located on X chromosome. Similarly, factor 8 hemophilia, again in human beings, is located on X chromosome. In the plant world, we don't have X-linked inheritance, good example. So, but these are two classic example जो आप NCERT में भी पढ़ते हो, right? Now, based on allelic combination, let's say that you are studying gene A and gene B, and if gene A is linked with gene B, both dominant, or gene A is linked with gene B, both recessive, okay, then this will be called cis linkage or coupling linkage. So it is just a matter of chance. There is a long chromosome and A is linked with dominant copy of B. Simple. They are cis linked or they are coupling linked. But it is not, right? What is written after somatic function? This is my abbreviation for function. Okay. And what is translinkage? Let's say that there are two genes, E and F. So what if dominant E is present with recessive of F on the same chromosome, or recessive E is present with dominant of F on the same chromosome? Then it will be called translinkage or repulsion linkage. So that's what. You are getting it now? So here coupling and repulsion is not being used as explanation for linkage, rather a type of linkage, and it is only a matter of chance. On the same chromosome, A, B, C can be all coupled and D can be representing repulsion. So on one chromosome, it keeps going on. Okay. What is a somatic function gene? Somatic function gene means any gene that controls any feature of your body except the determination of sex. So somatic function is everything related to your structure, your metabolism, everything if it is not related to determination of sex. So that is somatic function. So either you have somatic function genes or you have sexual function genes. Very limited sexual function genes which will determine the sex, which will provide the sexual differentiation and all. All other genes are somatic function genes. Your voice broke. Okay. Can you please repeat the abbreviation? This is function. Shraddha, this is function. I hope you got this now. All right. Now we come to the features of linkage. So genes that are close together on the same chromosome usually segregate as a unit, and therefore they get inherited together. So do not assort independently. Genes occasionally switch from one homologous to other through the process of crossing over, something that we have already discussed. Among the offsprings, the parental type individuals predominate. Non-parental types are less in number and they are produced by crossing over. They can affect autosomes. They can also affect sex chromosomes. We already know. And then we have just now seen on the previous page how test cross will be showing. Here we have taken the example of plant, but same can be seen in animals. So test cross in which plant is heterozygous for two completely linked genes yield only two types of progeny, each displaying one of the original combination of traits present in parental generation. On the other hand, independent assortment produces four types of progeny. So I hope this feature was already clear. So these are the salient features. Going back. So now we are understanding the types also and characteristic features also and detection also. We are now left with these two things: significance, impact on inheritance. We very well know what is the impact on inheritance. It causes the deviation from Mendelian ratios, I'm going to come to that. Let's go to the next concept now. Now we are here. Then we go to impact. Impact on inheritance. So first one is deviation from Mendelian dihybrid and trihybrid ratios. That is the first one. No independent assortment. And you can write the reason also, that they are located on the same chromosome, and how meiosis will be bringing them together, right? Then parental types predominate in F2. Then suppression of recombinants in F2. So this is much more than 9 और 1 के कंपैरिजन में बहुत ज़्यादा होगा, and this is much less than 9331. I hope you get this point कि recombinant क्यों suppress होंगे. Then moving further, recombinants may be 0 in complete linkage. If there is complete linkage, the number of recombinants is going to be 0. And test cross, if there is complete linkage, then only 2 phenotypes are there, right? And both are parental. Then it leads to 2 or more traits inherited together, right? So these traits can be pretty beneficial traits some of the times, and that ways it is helpful in crop improvement program, cattle improvement program, and sometimes the very same thing can be harmful. Do I give you a very interesting example? Half of the people, okay, half of the people who get premature baldness, half of the people, right, who get premature baldness, they also tend to develop hypertension later in their life because these two genes are tightly linked in recessive form. So these are the points that you will be mentioning about the impact on inheritance. और सबको दो दो सेंटेंस में आप इलेबोरेट करोगे तो इट इज इटसेल्फ एन आंसर विच मोस्ट ऑफ योर कॉम्पिटिटर्स विल फाइंड वेरी हार्ड टू ऑर्गेनाइज दिस इंफॉर्मेशन. जबकि इट इज वेरी ऑब्वियस बट यू नीड टू ऑर्गेनाइज दिस इंफॉर्मेशन टू राइट दिस आंसर. सो दैट इज इम्पैक्ट ऑन इनहेरिटेंस. ऑलराइट, सो ये हो गया हमारा इम्पैक्ट ऑन इनहेरिटेंस. एंड लास्ट, इफेक्ट ऑफ क्रॉसिंग ओवर. So crossing over results into recombination. Give me a moment. Results into recombination. Results into recombination. It breaks up the association of genes that are close together on the same chromosome. Processes as opposite effect. Linkage tends to inherit two different genes in the same allelic combination. Crossing over will break that allelic combination. So linkage keeps particular genes together and crossing over often mixes them up. And frequency of crossing over between two linked genes is mostly directly proportionate to the distance between the genes. And later on, now it seems it will happen tomorrow, we are going to see how crossing over frequency is used to essentially determine the distance between the genes. Because what we have just now learned, that frequency of crossing over between two linked genes is mostly directly proportionate to the distance between the genes. All right, and then you have got this one. So if there is no crossing over, right, let's say homologous chromosome pair is there, right, dominant A, dominant B together, recessive A, recessive B together. So in meiosis 2, after meiosis 2, they all have separated in parental type, and all resulting chromosomes in gamete have original allelic combination which are just like parent, that is non-recombinants. Avnish asking, does it lead to evolution? This will be too early to jump there. Yes, to an extent, crossing over increases variations, and variations will lead to evolution. But there are many other sources of variation, Avnish. Okay. Now, what if the crossing over takes place? So crossing over may take place here. So notice these two chromatids are going through crossing over. And if that happens, notice here, parental type non-recombinant. This is also parental type non-recombinant, but do you notice here? Recombinant. So in this case, half of the resulting gametes will have unchanged chromosomes, but other half will have recombinant chromosomes. So in one particular meiosis, if between A and B crossing over happens, A and B combination will be broken. But is it necessary that A and B crossing over will happen in every single cell that is going through meiosis? Answer is no. Some of the cells will have, rest of the cells won't have. And that way you will never get the Mendelian ratio. So that is the effect of crossing over and I would say linkage. So if there is linkage, it can be broken by crossing over. Vivek is asking, sir, how crossing over is different from mutations? बस mutation परसों ही पढ़ने जा रहे हैं. It will be very clear. Recombination is crossing over is a type of recombination. In recombination, always there is going to be two DNA which interact with each other. In mutation, two DNA don't interact with each other, but there may be some error in replication, some chemicals, some radiations, which will change the nucleotide sequence. But there is no interaction among the DNA. There is no exchange or transfer of genetic information from one DNA to another. So when two DNA molecules interact, and if there is an exchange or transfer of genetic information from one to another, this is something known as recombination. Crossing over is a type of recombination. But if you ask about how mutations take place, so error of replication, sometimes some chemicals, some radiation, and there is no exchange of information between DNA molecules. So I hope Vivek, abhi ke liye itna kafi hoga जानना कि how mutations are different, and when we see mutation happening, अभी हमें क्रॉसिंग ओवर भी करना है, म्यूटेशन उसके बाद वाली क्लास में होगा, तो you will be amply clear about it. So now we are knowing all these things what we were supposed to know. Okay, so give me a moment about linkage. All right, so we have seen significance and impact on inheritance also. I've given you all those points here, and relation with crossing over also. So 4 points to explain, 2 points, like 4 points worth of diagram. And now for the paucity of time, we will have to leave it here, and tomorrow, equipped with this insight, we will get into linkage mapping. But looks like only 4 parts, but it will take around the same amount of time. So it was a little optimistic calculation that today we could be doing both. मैं चाहता था कि कर लूं, but not at the expense of running through the conceptual clarity. Therefore, it's okay. In previous batches also, this much time has been taken by linkage and linkage mapping. But it is one of the important topics, so it is more important to understand it nicely rather than rushing through. Rest of the topics are going to be much more simpler, so you will not be taking the same amount of time. So that's all when it comes to the content part. Now all your questions are welcome.